Scientists have captured on video the intracellular version of a postal delivery service. Reporting in the journal Biochemical and Biophysical Research Communications, the bioengineering researchers at UC San Diego published time-lapse images of a message-carrying protein called pexillin. They labeled pexillin with a red fluorescence marker to make it stand out in live cells. In the reaction of those cells to an external stimulus, pexillin moved abruptly from hubs of communication and transportation activity toward the nucleus.
It's astonishing to us who thought the cell was very simple before, but it's really very, very complex. And I'm not sure we're covering everything as yet. And certainly we do not know all the interactions among these molecules to bring this whole cell into action, in motion or in other activities.
Chen and the paper's co-author, Yingli Hu, filmed the red pexillin molecules moving in tandem with green fluorescence-tagged traces of cytoskeleton. Scientists have confirmed over the last 10 years that higher organisms use pexillin as a transmitter of locomotion and other signals. They were simply unable to actually see the process occur.
But the availability of these labels that we can put in the cell together with particular proteins, in our case pexillin and actin, but you can do many other kinds of proteins, and then we can see the dynamics, how these proteins move, how do these molecules move inside the cell with time and in space. So it's a three-dimensional plus the time, you can call it a four-dimensional movement that is made possible by this live cell video microscopy.
Pexillin is also intimately involved in transmitting signals from several classes of growth factor receptors to the nucleus. Researchers are eager to understand these interactions because their malfunctions have been linked to a variety of cancers, tumor metastasis, and other disease processes. Tumor-causing versions of signaling molecules may attach to pexillin. This may disturb the normal adhesion and growth factor signaling steps required for controlled proliferation and cell growth. For example, human papillomavirus makes a protein that binds to pexillin. That interaction may contribute to the potential of this sexually transmitted virus to cause cervical cancer. In their study, Chen and Hu obtained cells from the intermembranous lining of a cow aorta. They added genetically engineered proteins tagged with red and green fluorescence markers to the bovine aortic endothelial cells and grew them in tissue culture.
In this cell, there is a flow of fluid over the cell from your right to your left, and the pexillin is moving along the direction of the flow. In this motion of the red labeled pexillin is alongside the green labeled actin filaments, as if the pexillin is using the active filaments as a track in their migration from the perimeter to toward the nucleus. By showing they're moving together, at least we have some insight into the possibility of how pexillin can transmit information from one region of the cell, such as the periphery, to another region, such as the nucleus.
Chen, Hu, and their colleagues at UCSD had previously reported that pexillin can rapidly assemble and disassemble.
Well, just like our walking is almost the same.
Right.
As you walk, you have to lift your foot here, sole, but you have to step down on the front. The forces are very different. And then for the next step, you again, go like this. So the same is true here. So those focal adhesions, they will be disassembled in the back, just like our sole. And front has to be a little bit actually tighter. Right. And then the next moment it cycles back. So that's how they move. Actually, Indy has shown that when we lift up this, the cell actually, the whole cell moves forward. Some of the pexillin, they stay here firm as the cell passes through them. Until the cell passes through them, they then lift.
Researchers are applying the fluorescence labeling technique to the many other signaling and structural proteins at focal adhesions.
When we piece all these things together, then we can get a much more complete understanding of how the cell functions, both in migration and in signal transduction when you try to change the gene expression. So these are very important processes. If we understand the details of these, then not just we understand how the normal cells function, but we can also look at disease cells to see why sometimes they don't move properly, or why they don't transmit the information properly and have abnormal gene expression.